Thank you, Dr. Stetz. And thank you for the opportunity to share our work. A lot of the work I'm showing today is in close collaboration with computational biologist Kevin Weehee. So determination of the paired antibody repertoire at great depth is required for understanding adaptive immunity as well as for antibody discovery, and in particular in our work at understanding the antibody response to HIV infection and HIV vaccination. One challenge is that the variable domains of the antibody heavy and light chain genes reside on separate mRNA transcripts, making the paired analysis of these antibody genes in high throughput challenging. And so current techniques involve characterization of the heavy and light chain repertoire separately using next generation sequencing or together, but only able to analyze small numbers of B cells at one time. So to overcome this problem, we're utilizing a flow focusing apparatus coupled with oil droplet PCR that was developed by the Georgiou laboratory at the University of Texas. And so using this apparatus, we're able to profile the paired antibody repertoire of as many as 6 million B cells at one time. And so in theory, we can use not just B cells, but any type of immune cells or cells. But for our purposes, we usually perform a B cell enrichment step using either magnetic beads or flow cytometry. And in our case, most of our samples are a single vial of PBMCs, about 10 million, so we can recover about anywhere between 100,000 to 600,000 B cells that we can then profile using this technique. So shown here and on the next few slides is a schematic of how the instrument works. So the flow focusing apparatus encapsulates single B cells into individual oil emulsion droplets using an aqueous phase solution and an oil phase solution. So shown here is a cartoon of the nozzle where all the magic happens. And so these are where the oil droplets are formed. So the nozzle consists of three separate chambers. The first is a hypodermic needle, which contains your cells, in this case B cells, with PBS. The second chamber consists of a cell lysis buffer, as well as oligo-DT beads, and so these beads will capture the mRNA from the cells. And then the outer chamber consists of oil to form the droplets. So by regulating the nozzle outlet size, as well as the speed of the aqueous and oil phases, we can create uniform oil droplets that encapsulate one cell and about 15 of these magnetic oligo-DT beads. And the advantage is these droplets are stable enough to collect in bulk fractionation, so we just collect them in different tubes, and so this allows us to examine millions of B cells at one time. So once the cell is in the oil droplet, the lysis buffer lyses the B cell, and then the mRNA of the BCR heavy and light chain genes are captured onto the beads. And so we use primers that then bind to the constant regions of the heavy and light chains to perform reverse transcription. And then we amplify the cDNA with overlapping PCR primers that physically link the heavy and light chain variable regions. And so since the heavy and light chain genes are now linked onto one amplicon, we can then disrupt the oil droplets and prepare libraries for next generation sequencing in bulk. So we initiated our studies by performing paired repertoire sequencing analysis from 3 individuals in our CHAVI cohort. 2 of the infected individuals, CH848 and CH0219, were previously identified to produce broadly neutralizing antibodies. And then we also did the paired analysis from a single seronegative individual, CH1293. So shown here is a bar graph of the heavy chain gene family usage by clonal lineage members from, in this case, the HIV-infected individual CH848. You can see the B cell clones are in black, and then the number of clonal members or the number of sequences recovered are shown by the light gray bars. And so I just wanted to demonstrate that we can detect B cell clonal lineage members from diverse gene families, and that they also contain certain clones that have expanded to include multiple B cell clonal lineage members, as evidenced by having a high gray bar compared to the black bar. So the same observation is the case for the other infected patient, CH0219, and for the seronegative individual. And so I went quickly, but we do see differences in gene family usage between these 3 individuals, but as of yet, we don't have enough samples to determine if this is a significant biological variation. So many HIV broadly neutralizing antibodies, they have long HCDR3 regions, as well as a high degree of somatic hypermutation. So shown here are dot plots of the clonal HCDR3 amino acid links on the left and the average clonal somatic hypermutation on the right for all of the three individuals we did the paired analysis. So you can see that we get clones with a range of HCDR3 links, with some clones having HCDR3 links rather long, greater than 20 amino acids. And on the right, we can get clones that are very close to germline with little somatic hypermutation, as well as clonal lineages that have undergone some substantial somatic hypermutation, approaching 20%. So we focus our initial studies, at least our paired studies, on a well-characterized class of HIV broadly neutralizing antibodies known as the VRCL1 class. And so cross-donor analysis revealed that multiple HIV-infected individuals could produce a class of antibodies that structurally mimic CD4, and these antibodies therefore can potently neutralize a majority of circulating HIV strains. And so this class is referred to as the VRCL1 class, named after one of the first antibodies isolated in this class. And so VRCL1 class antibody receptor genes, they have distinct genetic characteristics. One, they have high levels of somatic hypermutation, but they also have restricted heavy chain gene usage, utilizing VHEVI1-2 or VHEVI1-46 heavy chain genes paired with light chain genes that have an LCDR3 length of a short LCDR3 of only 5 amino acids, and usually V-kappa 320 families. And so it's been previously shown that only around 1% of human light chains have these LCDR3 links of 5 in the repertoire, so getting this genetic pairing is expected to be rare. And so interestingly, we looked in patient CHA48, and you probably don't remember because I showed it too fast, but we did observe a clonal expansion of VHEVI1-2 heavy chain gene families. And so given these results, and since we had paired analysis with this technology, we looked for VHEVI1-2 heavy chains paired with kappa 320s that had an LCDR3 length of 5 amino acids in CH848 that would be identical to this VRCL1 class genetics. And so we found 579 paired antibody sequences that matched the VRCL1 class genetics in patient CH848. Shown here in the top table are the top 6 clones with the most members. And you can see to the right of the table are the minimum, median, and maximum mutation frequency that was observed for each of these clones. So you can see that each of these clones contain members that were substantially divergent from germline, indicating that antibody maturation has happened. On the bottom is just a phylogenetic tree of the most abundant clone, demonstrating that large clonal expansion of these VRCL1-like antibodies in patient 848 that vary in divergence from the germline with unmutated common ancestor shown at the very left there. So next, we selected 2 antibodies from each of the top 5 most abundant clones in CH848 to produce as recombinant antibodies. We then tested these antibodies for binding in an ELISA assay and neutralization in the TZMBL neutralization assay. And so out of those 10 antibodies that we selected, 7 of them have yielded recombinant antibody thus far. So shown here is antibody binding to the CH848 transmitted founder envelope on the left, and a resurfaced core ONV protein named RSE3 on the right, measured by ELISA. So VRCL1 as the positive control bound to both of these ONV proteins, shown by the line with the blue triangles. But we did not observe strong binding for any of the 7 VRCL1-like antibodies tested in patient 848. And additionally, these VRCL1-like antibodies did not neutralize in the TZMBL assay. And so this determined that just having this rare germline pairing of VHEVI1-2 with a light chain of LCDR3 is not enough to recapitulate the VRCL1 binding and neutralization of HIV, and therefore specific somatic mutations that VRCL1 has are required for this activity. So shown here is an amino acid sequence alignment with VRCL1 and an antibody from 848 that has the closest relation to VRCL1 labeled VCL1. The heavy chain at the top, there are green boxes that show residues that have been demonstrated to be important for VRCL1 binding. And you can see that these 3 residues are encoded in the germline of VHEVI1-2, and they're maintained in the antibody VCL1 from patient 848. And that's partially a reason for this restricted germline usage, is some of the contact sites for VRCL1 are encoded in this germline gene. But conversely, in the light chain, there are 3 regions of mutation that are important for VRCL1 activity that VCL1 does not have. First, there's a 3 amino acid deletion in the CDR1, a tryptophan mutation downstream of the CDR2, and a glutamate mutation in the LCDR3. And all of these have been previously shown to be critical for VRCL1 binding and neutralization. So what we did is we engineered these mutations into a VCL1 clonal lineage member and then tested, did that improve binding? So on the left is binding of VCL13, as well as VCL13 with a single G to E mutation that was the mutation in the LCDR3, and then VCL13 with all three of those light chain mutations shown on the previous slide labeled trimute. VRCL1 binding is shown in purple, and you can see that neither the single or triple light chain mutations were able to convert VCL13 into an ONV binding antibody. Now, to rule out contribution from the heavy chain, on the right side, we paired the mature VRCL1 heavy chain with each of the light chains from VCL13. And you can see that VRCL1 paired with its native light chain shows strong binding by the purple line on the graph on the right. However, VRCL1 heavy chain paired with either mutated VCL13 light chains do not bind. So these data demonstrate that mutations other than the three we tested just in the light chain are required for VRCL1 activity, and it will be important to identify what all these specific mutations are that are required for VRCL1 binding and neutralization in order to target generation of these antibodies by an HIV vaccine. So in summary, we've established the technology to be able to profile the paired antibody repertoire at high throughput. We've sequenced the paired repertoire of 2 HIV-infected individuals that produce broadly neutralizing antibodies, as well as a seronegative individual. And repertoire sequencing analysis identified a distribution of mutated antibodies with diverse HCDR3 links. In addition, we identified antibodies with the rare genetic characteristics of the VRCL1 class bnAbs in patient 848. And although these antibodies utilize the germline antibody gene pairing of VRCL1, they did not bind or neutralize. So this indicates that there is a complex maturation pathway that will be required to be elicited if we want to elicit VRCL1 class antibodies by vaccination. So in conclusion, high throughput profiling of the paired antibody repertoire is possible with high accuracy. An analysis of critical mutations in VRCL1 that are necessary for neutralization are underway. And additionally, in both infected individuals, CH848 and 0219, we have identified more mutated members of the bnAb lineages that were previously found, and they're being characterized. So I'd like to end by thanking the many individuals that are and have contributed to this work, as well as the funding sources that make this work possible, including a CFAR small grant. And thank you. I'll take any questions.
Any questions for Todd?
So, so congratulations on the really nice work. It's lots of work in there. So for your new technology with the paired antibodies, do you sequence the complete with heavy light chain variable region now?
Yeah, no. So if you use, so when they're linked, it's about 1,200 base pairs long. So if you use the Illumina platform, we can sequence most of the V gene to get the CDR3 as well as the gene call. But if we want to make the antibodies, like in our case, we have to then prepare 2 other libraries of the full heavy chain and the full light chain if we're sequencing by Illumina. If you use sequencing like PacBio or something that can sequence longer, you can do that all in one read. But for our purposes, we're making 3 libraries, a paired library, a heavy chain library, and a light chain library.
That's good. So that we can function the study. But the next question is like, have you tried to do the repeated run like your other way? So you can make sure so some of the variants are actually can be identified in 2 separate runs with your paired technology.
Yeah, so we actually do, I didn't discuss, we do replicates after the first PCR, but since the RT and PCR occurs, the first steps occur in the oil droplet with the B cell in it, it's difficult to have a technical replicate of that PCR amplification step. So we acknowledge that some of those sequences will contain some errors.
Last question. So you said VCR01 is a very long CDR3. It's a very unique. Is that right? Or VCR01? No.
So VRCL1, I mean, they don't have a very long heavy chain CDR3, but the light chain CDR3 is very short, and it's restricted to having only 5 amino acids.